Good afternoon, students. Today we are going to discuss a very new topic, that is lipid metabolism. So these are the topics we are dealing with. That is introduction. Next is beta oxidation of fatty acids, ketogenesis, ketolysis, and final one is ketoacidosis. So introduction. Lipids. I think you are all familiar with lipids, right? So lipids is very important, or it is very essential for cell structure and their functions. This is mainly due to their nature, that is they having hydrophobic as well as non-polar nature. Hydrophobic means tendency to mix with the water, and non-polar means there will be no charge separation. So among our human weight, 15 to 20% is lipids. Triacylglycerols, they are very important lipids and they constitute 85 to 90%. And these body lipids, they are stored in adipose tissue. And this lipids is differ from other body components like amino acids, fatty acids. They are very different and they are unique in action. We cannot store carbohydrates and proteins, but lipids can be stored, and they are also act as an insulating material in case of animals for maintaining their body temperature. So next is very important cycle, that is beta oxidation of saturated fatty acid or palmitic acid. So beta oxidation, what is beta oxidation? Beta oxidation means the oxidation of fatty acids using beta carbon atom, that is called beta oxidation. So in our body, most of the fatty acids, they are oxidized by the beta oxidation. So there are three steps involved in this beta oxidation. One is activation of fatty acids, that is takes place in cytosol. Next is transportation of fatty acid to mitochondria. And final step is beta oxidation, that is proper, that takes place in mitochondrial matrix. In beta oxidation, actually the breakdown of fatty acid to acetyl-CoA. Then this acetyl-CoA will enter to citric acid cycle, and this is aerobic reaction. And this will take place in kidney, heart, liver, etc. In brain will not take places. So let's start with our steps. First one is activation of fatty acids in cytosol. So the fatty acid is reacting with thiokinase and we are getting acyl adenylate. So in between there is ATP. We are converting into pyrophosphate, and this pyrophosphate is reacting with pyrophosphatase to form phosphate. Then this adenyl cyclate react with a coenzyme and they form AMP, and they results in acyl-CoA. So this is the activation of a fatty acids in cytosol. And the next step, that is the transportation of fatty acids to mitochondria. So this reaction taking place in mitochondria. So inner mitochondrial is impermeable to these fatty acids. So we are using a special transport system, that is carnitine carrier system. So here our reactions takes places. So this also takes place by the 4 steps. And first one, see acyl-CoA. You can see here acyl-CoA. Acyl group of acyl-CoA, that is transferred to carnitine. And this reaction is catalyzed by, that is carnitine acyl transferase. And finally, we are getting acyl carnitine with the help of coenzyme A. And this acyl carnitine that is transported to a membrane using a carrier protein. So in center you can see carrier protein, that is inner mitochondrial membrane, and they'll reach to mitochondrial membrane. So acyl carnitine, and this acyl carnitine will react with this carnitine acyl transferase 2 with the help of coenzyme A, and we are getting acyl CoA. So this process will take place repeatedly, and this acyl CoA again converted into carnitine using carnitine acyl transferase 2, and they will go to cytosol. So this process keeps on repeating. So first here acyl CoA is transferred to carnitine using this enzyme, that is transferase 1, also with the help of coenzyme. Then finally we are getting acyl carnitine, and this acyl carnitine using carrier protein, they are transferred to mitochondrial matrix. And this one is acyl carnitine again reacting with a transferase 2 and help of coenzyme A, we are getting acyl CoA, and the same process repeats. So this is the explanation of this process, step 2. Next is third step, that is very important step, beta oxidation proper in mitochondrial matrix. So here you can see we start with acyl CoA. So acyl CoA will react with this enzyme, that is acyl CoA dehydrogenase, and we are getting trans delta square enol CoA. So in between a reaction takes place, that is FAD is converted into FADH2, that is oxidation, and so we got trans delta square enol CoA, and this will react with enol CoA hydratase and results in 3L hydroxy acyl CoA. In between, hydration occurs, hydration by hydratase. In first step you can see oxidation, and second step you can see hydration. So we are using enzyme is enol CoA hydratase, and we are getting 3L hydroxy acyl CoA, and using dehydrogenase we are getting beta keto acyl CoA. Again oxidation takes place, that is NAD positive is converted into NADH plus H positive. NAD positive means nicotinamide adenine dinucleotide. FAD means flavin adenine dinucleotide. I think you all studied these things in enzyme chapter. So using this hydrogenase enzyme we are getting beta keto acyl CoA. Then this acyl CoA will react with beta keto thiolase, and here the cleavage occurs, that is two parts, that is fatty acyl CoA plus acyl CoA. The cleavage occurs by this thiolase enzyme. So in first step you can see oxidation, next is hydration, again oxidation, and last step is cleavage, and they results in two acids, that is two fatty acids, that is fatty acyl CoA and acyl CoA. So this release of acyl CoA, they will shortens 2 carbon atom. Actually this acyl CoA is a substrate for the next round of oxidation, and this process will continue until all carbon atoms will converted into acyl CoA. So this is the third step of the beta oxidation. Next is overall reaction. So from third step you will get this. First we are using acyl CoA, then FAD, then NAD positive, then hydration. We are using coenzymes. So already I told you that by the formation of acyl CoA we are shortened 2 carbon atoms, that is N minus 2 acyl CoA plus acetyl CoA. Then oxidation takes place, so we are getting FADH2. Again oxidation, that's why NADH plus H positive. In the case of palmitoyl CoA, this is palmitoyl CoA. Means the process is all same. Instead of acyl CoA, we are using palmitoyl CoA. That's it. So in beta oxidation of palmitoyl CoA, there will be 7 cycles of beta oxidation, and they results in 8 acetyl CoA. So this acetyl CoA will enter the TCA cycle or citric acid cycle, and they get oxidized to carbon dioxide and water. So this is the overall reaction. Next is energetics. This is the palmitate standard energy. It is 2340 calorie. Then by oxidation process we are yielding 129 ATP. Next is ketone bodies. What is ketone bodies? Ketone bodies means acetone is a ketone body. Acetoacetate is a ketone body. Beta hydroxybutyrate is ketone bodies. Ketone bodies are water soluble, also energy yielding. So next is ketogenesis. So ketogenesis means formation of ketone bodies. In liver, synthesis will take place. Ketone body synthesis takes place in liver, and the enzyme for this process is located in mitochondrial matrix. Here also we are using acetyl CoA. So let's see the cycle. We are starting with 2 acetyl CoA. Acetyl CoA will react with thiolase and coenzyme A. We are getting acetoacetyl CoA, and this acetoacetyl CoA will react with HMG CoA synthase. HMG CoA synthase, and we are getting beta hydroxy beta methyl glutarate CoA, that is HMG CoA. In between, acetyl CoA is getting converted into coenzyme A. So in first step we are only using coenzyme A. In second step, acetyl CoA is converted into coenzyme A. Next, this HMG CoA is reacting with HMG CoA lyse, also with acetyl CoA, and we are getting acetoacetate. So this acetoacetate undergo 2 reactions. One is decarboxylation. So while undergoing decarboxylation, we are getting acetone, and while undergoing reduction, we are getting beta hydroxybutyrate. So these are the products of the ketogenesis, acetone and beta hydroxybutyrate. So we are start with 2-acetyl-CoA and we are ending with acetone and beta hydroxybutyrate. So this is ketogenesis. And what are the enzymes we are using here? First one is thiolase, second one is HMG-CoA synthase, and third one is HMG-CoA lysis, and last step is decarboxylation and reduction. So this is ketogenesis. Next is ketolysis. That is beta hydroxybutyrate. Ketogenesis is ends with this product, that is beta hydroxybutyrate. So ketolysis is opposite of ketogenesis. Same processes takes place in. So enzymes is a little bit different. First we are starting with hydroxybutyrate and with the help of dehydrogenase enzyme and also NADH, there is oxidation takes places, we are getting acetoacetate. And again this acetoacetate is reacting with 3 ketosur-CoA transferase. And we are getting acetoacetyl-CoA. In between, succinyl-CoA is converted into succinate. And this acetyl-CoA will react with thiolase, also with coenzyme A. And we are getting 2-acetyl-CoA. So in ketogenesis, we are starting with 2-acetyl-CoA and ending with 3-hydroxybutyrate and acetone. In ketolysis, we are starting with 3-hydroxybutyrate and we are entering with 2-acetyl-CoA. Means same processes takes place in. That is ketolysis. Means we have already discussed that these ketone bodies are water soluble. So water soluble, so they are easily transferred into liver. This acetoacetate and beta hydroxybutyrate, they are very important ketone bodies. They produces more energy. So in mitochondria, ketone bodies utilization will now take places. Then production of ketone bodies and their utilization is very important. If the glucose level is low in our body, ketone bodies will means their role will take places, especially in the case of starvation and diabetes mellitus. That is, in starvation, glucose will be low in our body because we are not taking any food. So ketone bodies is a good source for our brain and nervous system. So 50 to 70% of brain needs takes place by these ketone bodies. Then so the ketone bodies is more produced in liver and they are utilized by the extra hepatic tissues. So the concentration of ketone bodies is, that is 1 mg per dl. And the ketone bodies is excreted through urine. It cannot be excreted through the urine, but it is very low. So this can be dictated by a test, that is Rother's test. The ketone bodies synthesis is more, but their utilization is less. So in that condition, the concentration in the blood will increases. Synthesis is more, but utilization is less. So in that case, the concentration will increase in blood. So that condition we can call as ketonemia. Then ketonuria means excretion of the ketone bodies in the urine. So the overall picture of ketonemia and ketonuria, we can call it as ketoacidosis. So sorry, ketosis, not ketoacidosis, that is ketosis. Ketonuria means excretion of the ketone bodies through urine. And ketonemia means synthesis will be more, but their utilization will be very less. So in that condition, the concentration will increases in the blood. So that is called ketonemia. Next is starvation. Starvation means it is mainly due to the degradation of fatty acids. So we are not having food, so we are not getting any energy. So the fatty acids will degrade. So when the fatty acid will degrade, this results in the more production of the acetyl-CoA. And this process is handled by the citric acid. So if more acetyl-CoA is released, there will be deficiency in oxaloacetate in citric acid cycle, and it results in the glucose synthesis. So this will help our daily requirements for the brain. So finally, the acetyl-CoA will accumulate, and it results in the more production of the ketone bodies. That is starvation. So during starvation, this will take places. First, the fatty acids will degrade, and this results in the acetyl-CoA overproduction. So this is handled by citric acid cycle. So when acetyl-CoA is getting more, oxaloacetate will be low in citric acid cycle. So this results in the glucose synthesis, and they help in our daily needs. So as a result, acetyl-CoA will get accumulated, and this results in the more production of ketone bodies. Next is diabetes mellitus. Diabetes mellitus means insulin deficiency. This is mainly due to increased lipolysis as well as impaired, that is proper metabolism, improper metabolism of the carbohydrates. And this results in the accumulation of acetyl-CoA, and finally the ketone bodies will form. That is diabetes mellitus. Next is ketoacidosis. Ketoacidosis means the ketone bodies will increase in our blood, and they results in ketoacidosis. Here, carboxyl group, that is, I told you that important ketone bodies is acetoacetate and beta hydroxybutyrate. So these both acids having carboxyl group. So their pKa is around 4. So here the ketone bodies will disassociate, and they release more hydrogen atoms. So this will lowers the pH. So the plasma volume will reduce in our body due to dehydration, and they results in the extraction of the glucose as well as ketone bodies. So that condition is ketoacidosis. Diabetic ketoacidosis is very dangerous. Sometimes it results in coma, even death also. So again I am repeating, in ketoacidosis, We have already discussed that acetoacetate and beta hydroxybutyrate are ketone bodies. So they are very strong ketone bodies. In that you can see carboxyl group. So their pKa is around 4. So these ketone bodies, that is acetoacetate and beta hydroxybutyrate, in our blood it will get disassociate and they will release more hydrogen atoms. So this release of hydrogen atoms will lower the pH. So when pH lowers, the plasma volume will get reduced. This is because of dehydration. And finally, they result in the excretion of the glucose and ketone bodies. And treatment of the ketoacidosis. That means proper food. That is very good remedy. And second one is administration of insulin. So this is the main two remedies for the ketoacidosis. So that's for today. Thank you.